Everyone. This is Medical Microbiology Practical Lecture 4, College of Pharmacy, the third stage and sixth semester. I am Dr. Marwan Hussein. Today I'm going to explain and show you the culturing of Streptococcus on the blood agar. So blood agar, it is a general and selective media. So general media for all of the microorganisms. I mean, even fungi like Candida and bacteria can grow on the blood agar. So generally, it is better to use sheep blood agar, not a human blood agar. But we cannot now. It is sometimes it is difficult to get sheep blood and then adding to the blood agar and then auto living. So we use a human blood to do this. It's okay, of course. So the as I said, it is general media for most of bacteria, and it is selective media for some gram-positive cocci. It's a selective media. Why it is selective media? Because a gram-positive cocci, Streptococci especially, and Staphylococci. So today we're going to talk and do and work in Streptococcus. Streptococci produce exotoxins, and these exotoxins destroy red blood cells, RBC, and hemoglobin. So it is hemolysis, the name of toxin. It is hemolytic bacteria. So the ability of these bacteria to produce, Streptococci, to produce this enzyme and destroy, this is make blood agar as a selective media for them. The principle, as I said, according to hemolytic activity, we classify a groups of bacteria of Streptococci to categories, to classes. The hemolytic activity, according to the hemolytic activity, and to serological classification of Landsefeld. So our practical today is do the according to hemolytic activity, of course. Basing to hemolytic activity, we have three types of hemolysis bacteria: beta and alpha and gamma. So beta hemolysis, you will see the complete destruction of RBC, complete of RBC and hemoglobin. So it will appear as a clearing around the colony, clearing zone, and the color is like yellow around the colonies. But alpha hemolysis is partial destruction of RBC. It's incomplete destruction, incomplete, and produce, of course, a greenish discoloration around the colony and on blood agar, While the gamma hemolysis is non-hemolysis, and appear as a simple growth, just growing on the media. So according to the procedure today, you have a group, of course. You will and every group will work and do inoculation the sample of Streptococci on a blood agar. You have a blood agar on the bench, and you have the sample, of course, and you will do the streaking and inoculation, on the surface of blood agar, and then, of course, waiting for, as you have learned techniques of streaking and inoculation, and the next day you will have the results, and to see which group, which hemolytic, alpha, beta, or gamma, and then of course you will write the reports. I mean, the incubation will be in the, as you know, it is bacteria Streptococci 35 minus plus 2 centigrade for 24 hours. I mean, the next day you will see, you come to the lab and see the results. The number 2, look, sorry, this is the beta hemolysis. I'm going to show you this. How can you see after you do the inoculation? And this is the alpha. This is the beta. This is alpha hemolysis. This is greenish and incompleted. I mean, around the colony. And this is gamma hemolysis. So just grow on the blood agar without any clearing zone or without any hemolytic blood or RBC in the media. Another picture, of course. Sorry, another picture. Imaging to show you. The second thing, lab, you will do it. You have sample of Streptococci, but the another sample you will have from the throat. So among the student, you will do it. You can have the sample from your friend, and your friend have from you. By swab, you're going to use a swab, as you have learned before, from the throat, like this in the picture, and then by swab, you're going to do inoculation on the media, blood agar, I mean. So may it is grow, because Streptococci from the throat, it's a normal flora, if it is not pathogenic, so you will have just maybe it is green, growing on the media. But the pathogenic, it is hemolysis, of course. Or maybe you will find the hemolysis like beta or gamma from these samples which you have by your hand from your friends, of course. Of course, as I explained before, beta hemolysis is clearing around the growth, and it is completely hemolysis. Alpha, incomplete hemolysis, graining around the growth. It's partially, of course, hemolysis or destroying, but gamma, no changes in the media and no hemolysis, of course, so just growing on the media. This is another example. So Streptococci on the chocolate agar. Chocolate is like a blood agar. So if you have time, you're going to have in another lab, of course. Thank you for your listening.